Welcome everyone. In this video, we will gonna to discuss about the previous year questions of CUET-PG Life Science. So let's discuss about question number 21. The most stable structure for a random sequence DNA molecule under physiological condition is option 1 form of DNA, option 2 A form of DNA, option 3 Z form of DNA, option 4 B form of DNA. So the most stable structure of DNA is B form of DNA, and it is very easy to read it in or sequencing it in random sequence. We do what anyone for research will use the B form of DNA, and the B form of DNA is a right-handed DNA molecule. Now that Z form of DNA is a left-handed DNA structure. So you should have to know more about those DNA and their structure, because it's very important to know that what the DNA are looked like. So the B form of DNA is a right helical DNA structure, and Z form is a left-handed DNA structure. Okay, and A and C are also a right-handed DNA structure. But the most common or stable structure of DNA is a B form of DNA. Always remember that point. Question number 22. Porins are proteins found in gram-negative bacteria such as E. coli and allow dash to cross the outer membrane. Option 1 amphipathic solutes, option 2 hydrophobic solutes, option 3 non-polar solutes, option 4 polar solutes. So the correct answer for this question is 4 polar solutes. The porins or the protein which are found on the gram negative bacteria will allow the polar solutes to cross the outer membrane. Question number 23. In the enzyme ATP synthase, F0 domain is sensitive to option 1 oligophysin, option 2 oligosaccharide, option 3 oligomycin, option 4 oligonucleotide. So if you had read of about the etc, then you know what are the inhibitors for different complexes. Now this question is particularly asking about the inhibitor of a ATP synthase F0 domain. So they are very sensitive to the antibiotic, and the correct option for this question is option 3 oligomycin. Mycin is an antibiotic. Now so we choose the option 3 as a oligomycin is the correct answer, which inhibit the ATP synthesis process, and they will close the F0 site for binding of a hydrogen molecule. Okay, so the correct option is option 3. Question number 24. Which is not the type of positively charged amino acid? Option 1 arginine, option 2 histidine, option 3 asparagine, option 4 lysine. So the most common, there are only 3 positively charged amino acid: arginine, histidine and lysine. They are the positively charged amino acid because they have a basic group present on them, on their structure. Okay, now the asparagine, which is not a positively charged amino acid, and asparagine is a neutral amino acid because it is uncharged, so it's a neutral amino acid. So the correct option for this question is option 3 asparagine, which is not a positive charged amino acid. Except asparagine, all three are the common and the only example of the positive charged amino acid. So the correct answer is asparagine, which is not a positive charged amino acid, it's a neutral amino acid. So please remember all the amino acid and their types or the group they are in, either positive, negative or neutral amino acid. So learn them, because amino acid is the most common question for any competitive exam. Question number 25. In most higher eukaryotes, the fatty acid synthesis occurs in cytosol, but in plant it occurs in option 1 cytosol, option 2 endoplasmic reticulum, option 3 mitochondria, option 4 chloroplast. So the correct answer is chloroplast. In plants, that fatty acid synthesis occurs in chloroplast, and their degradation or their uh, breaking down will take in mitochondria. So the correct answer for this question is option 4 chloroplast, where the fatty acid synthesis will occur in case of plants, but in case of animals it will be in cytosol only. Okay. Question number 26. Microbes involved in biological fixation of nitrogen are A. Nitrosomonas, B. Azobacter, C. Pseudomonas, D. Anabina, E. Rhizobium. So, if you had read about the biological fixation by microbes of a nitrogen, you know that this all are the example of a nitrogen fixating microbes. Okay, so the correct option for this question is option 3, in which A, B, C, D, E all are the correct option. So I think that nitrogen fixation is the important topic. You should remember all the steps and the bacterias or enzymes which are taking part in that, and remember also about that which are the symbiotic microbes or the non or free living microbes and their examples also. Okay, so it is, I think, nitrogen fixation is the most important topic for all your competitive exams. So remember all the examples and their type, that either they are symbiotic or they are non or free living bacteria. So the in this question the correct answer is option 3, in which all these microbes are the uh, used for biological fixation of nitrogen. Okay. Question number 27. Match list 1 with list 2. In list 1 we had getting the phylum names, and in list 2 we will get their excretory organs. Now, if you remember about the most common one, that is arthropoda, and the arthropoda have the Malpighian tubules, then the correct option for C is 1. Now, platyhelminthes have the excretory body of a flame cells. Flame cells are mostly present in platyhelminthes. They are the flatworms which have flame cells in them, and that flame cell is their excretory organ. Annelida have the nephridia as the excretory organ, and mollusks have the feather-like gills. So the correct option for this question is option 3, in which all the options are given. You can match it and more learn about this phylum groups, because it's also an important question for your CUET-PG questions, okay, for life science questions. Question number 28. Match list 1 with list 2. So in list 1 we had gotten the types of amino acid according to their charged, uncharged basis. Okay, and in list 2 we get their examples, so we have to match them. Okay, now negatively charged amino acid. Now what are the negatively charged amino acid? If you had remember, there is common two example of negative charged amino acid. One is glutamic acid and the other is aspartic acid, also known as aspartate or glutamate. So for negatively charged amino acid, the correct answer is the option 3, negatively charged amino acid, and option 3 is aspartate. So 3 is correct match for A. Now B is polar uncharged amino acid. Now what are the examples of a polar and uncharged amino acid? Can anyone have examples? So please write in the comment box. So what are the examples of a polar and uncharged amino acid? There are mostly 5 amino acids which are polar but uncharged, and they are serine, threonine, asparagine, glutamine and cysteine. So in this list 2 we get only the example cysteine, which is 2. So the correct match for B is 2. Now come up on C, the aromatic amino acid. Now what are the examples of aromatic amino acid? So there are multiple examples of aromatic amino acids like tryptophan, tyrosine, phenylalanine. So in this list too, what we had gotten the phenylalanine. It's a aromatic amino acid because it have a benzene ring present on phenylalanine. So the correct match for C is 4. Now D, positively charged amino acid. So in previous question also we had discussed about the positively charged amino acid, and there are only 3 positive charged amino acid: lysine, arginine and histidine. In this question we had histidine. So the correct match for D is 1. And the correct option for this question is option 4, in which we will get all the matches. So the option 4 is the right answer for this question. And if you want to read about the type of amino acid and different type of charge basis amino acid, I had uh, made in the video on that topic also. You can watch from my YouTube channel, and I also had given in that description box. You can take the link from the description box and watch it, because it's a very important question. All the amino acid type questions are very important. They are most asked question from amino acid. Question number 29. Choose the vertebrates from the given list. A. Dogfish. B. Silverfish. C. Anogulia. D. Devilfish. E. Hippocampus. So the correct answer for this question is that option 3, in which A, C, D all are the vertebrates. That means dogfish, anogulia, and devilfish, hippocampus. All are the example of vertebrates except the silverfish. Silverfish is a kind of insect and but is known as silverfish. So don't get confused that it's a vertebrate. Silverfish is the example for the insect group. So it is not the vertebrate. So the correct answer for this question is option 3. Question number 30. Save tube elements found in angiosperms are A. Save tube, B. P protein present, C. P protein absent, D. Save plate, E. Companion cells. So the save tube elements found in angiosperms are save tube, save plate, companion cells and P protein present. So A, B, D, E all are present in the save tube elements which are found in angiosperm. So the correct answer or the option for this question is 1, option 1, A, B, D, E only. The P protein absent is not. Okay, P protein is present. So all the option except C are present or found in the angiosperm save tube elements. Okay. Thank you for watching. For more videos, you can like and subscribe to my YouTube channel.